Welcome to the SANS Science Experience, your source for the most current and impactful research and preprints in science. Before we dive into today's really mind-bending discovery, please take just a second to hit that like button and subscribe. It genuinely helps support us here at the SANS Science Experience. Let's just keep bringing you these insights. So today, we're embarking on a deep dive into an absolutely captivating corner of biology. We're talking about the intricate dance between aging, cellular health, and an enzyme you might have heard of, catalase.
Right.
And this isn't just simple chemistry. It's really about how a seemingly straightforward antioxidant is, well, kind of reshaping our whole understanding of longevity. Get ready to challenge some pretty deep-seated ideas.
That's exactly right. This deep dive, it's going to unpack a really groundbreaking study. It was done on fruit flies, Drosophila, and it uncovers this surprising, almost counterintuitive mechanism for, how lifespan might actually be extended.
Fruit flies again. They tell us so much.
They really do. So we're going to navigate some concepts that are maybe college-level science, but our goal is absolutely to make it engaging and clear for everyone, no matter your background. Think of us as your guides here.
Oh, wait. So our mission today, unpack the unexpected ways that redox regulation, that's that delicate balance of oxidation and reduction in our cells, and these critical cellular processes like autophagy play a huge role in how long an organism lives.
And importantly.
Yeah, and why this specific research forces us to totally rethink some, long-held theories about aging. It's a real scientific plot twist. Okay, let's unpack this scientific mystery then. For decades, it really felt like science had a clear villain in the aging story. Those reactive oxygen species, ROS.
The usual suspects.
Right. The oxidative damage theory of aging basically suggested that these ROS, just byproducts of metabolism, cause damage that builds up over time, leading to aging.
Makes sense on the surface.
It does. So the logical steps seemed obvious. To flood the system with antioxidants, like catalase, to neutralize these ROS. But here's the thing. Mostly, these attempts to extend lifespan just haven't panned out. They've been largely unsuccessful.
Yeah, it's been a bit disappointing on that front. And what's really fascinating here, and what this study really drives home, is that we often think of ROS, particularly hydrogen peroxide H2O2, as purely damaging, like cellular rust.
Right.
But H2O2 isn't just a bad guy. It's actually a major player in what we call redox signaling. Think of it like a tiny messenger molecule constantly talking within the cell.
Okay, so communication, not just damage.
Exactly. This balance, this redox homeostasis, it's about sophisticated internal communication. And the core, really surprising finding from this new study is that just overexpressing catalase, the enzyme that degrades H2O2, all through the fruit fly, robustly extends lifespan.
Wait, hang on. So more catalase breaking down this H2O2 messenger actually extends life. That already goes against a lot of previous findings, right?
It does. It's a significant departure.
How much longer are we talking?
Well, it's not insignificant. We're talking about extending both the median and the maximum lifespan in female flies by about 10 to 15%.
Okay, 10-15%, that's substantial. But only females.
Here's where it gets really interesting. Yes, only in females. This effect was not seen in male flies. And that's even though the males also showed really strong resistance to oxidative stress challenges.
Like what kind of challenges?
Things like giving them extra H2O2, or the herbicide paraquat, or even putting them in high oxygen hyperoxia. Their bodies handled the stress better, sure, but they didn't get that lifespan boost.
That's a massive clue, isn't it?
Yeah.
Suggests it's not just about mopping up damage. And it's definitely not universal.
Exactly.
So it brings up that question again. Why didn't older studies that also overexpress catalase find this? If the simple oxidative damage theory was right, it should have worked before, shouldn't it?
It should have. And the researchers tackled this head on. It looks like there are maybe two key reasons. First, the amount of catalase overexpression seems really important.
How much more do they get?
This study got a big jump, like a 5 to 10-fold increase in catalase protein. A lot of the older studies that found no effect only managed maybe a 1.75-fold increase. So sometimes you really need to push the system harder.
Okay, so dose matters. What's the second reason?
The second, and this is huge, is the sex specificity, that female-only effect. Many older studies might not have looked at sexes separately, or maybe just focused on males.
Ah, I see. So that difference could explain a lot, maybe even why some human anti-aging trials show mixed results between men and women.
It's definitely a possibility, yeah. A huge clue.
Okay, so here's where the plot twist gets even twistier. You'd think flies overexpressing this life-extending antioxidant would be super robust, right?
You would.
But the study found something else unexpected. These long-lived female flies, while great against oxidative stress, were surprisingly sensitive to complete starvation.
Right. Totally counterintuitive.
Yeah, like being strong in one way makes you weak in another seemingly unrelated way. How does that work?
Well, it points us directly to a really vital cellular process, autophagy.
Autophagy. Okay. The cell's recycling system.
Exactly. Often called self-eating, but in a good way. It's how cells clean house, recycle damaged parts, and generate energy when nutrients are scarce, like during starvation. It's fundamental for survival, and we know it plays a big role in longevity.
So what do they find with autophagy in these flies?
They found that these long-lived catalase overexpressors actually showed enhanced autophagy induction. Their recycling systems were ramped up.
Enhanced. How did they measure that?
They had clear evidence. They saw more lysotracker red dots, those glowy bits showing active recycling compartments, and they found higher levels of AG8 protein, both the starting form and the active lipidated form. AG8 is like the main marker for autophagosomes.
So all signs pointed to more autophagy happening.
A significant increase.
And crucially, this wasn't just happening alongside the longer lifespan. It was required for it, wasn't it?
Absolutely essential. They proved this pretty convincingly. When they blocked autophagy using something called AG5 RNAi, it completely wiped out the lifespan benefit from the extra catalase. Wow.
And treating the flies with chloroquine, which also inhibits autophagy, it caused that same weird starvation sensitivity.
Okay, let me try and wrap my head around this. An antioxidant makes flies live longer, but also makes them sensitive to starvation, and this whole benefit depends entirely on having more autophagy. It feels backwards.
It really does seem paradoxical at first glance, but the answer seems to lie in thinking about redox not just as damage control, but as a really finely tuned signaling system.
The messenger idea again.
Exactly. Imagine tiny chemical switches in the cell redox switches flipped by the local chemical environment. It's not changing genes, it's tweaking the proteins that are already there. Post-translational modification.
Okay.
And using a technique called redox proteomics, OxiCat, they found something really startling. Even though catalase overexpression reduced overall ROS, it unexpectedly led to an oxidizing shift in the general cysteine redox state in the female flies as they got older.
Hold on. An antioxidant causes an oxidizing shift. How on earth does that work? Less H2O2 leads to more oxidation. That sounds completely contradictory. It does, but it's brilliant when you see the proposed mechanism. By efficiently getting rid of cytoplasmic H2O2, the extra catalase actually interferes with the normal physiological H2O2 signaling.
It's silencing the messenger.
Precisely. That normal H2O2 chatter usually helps turn on other antioxidant systems, like the KEAP1 NRF2 pathway, a major protector. But in these long-lived catalase flies, that pathway wasn't getting activated with age like it normally would.
Okay.
So interfering with that normal H2O2 message ultimately leads to this overall oxidizing shift. They even saw lower levels of glutathione, another key antioxidant.
The implication is this internal oxidizing shift. It's not actually damage here. It's being read by the cell as something else.
Exactly. It's perceived by the cell as an internal state of starvation, like a false signal.
Wow. So the cell thinks it's starving, even when it's not, because of this weird redox shift caused by the antioxidant.
That seems to be the idea, and that false starvation signal is what powerfully triggers autophagy as a protective response, which, it turns out, is also pro-longevity.
It's like tricking the cell into running a survival drill, making it stronger.
That's a great way to put it, which obviously leads to the next question. How does this oxidizing shift specifically control autophagy to make the flies live longer?
Right. What's the actual switch?
This study points right at a protein called ATG4A. It's a major player in autophagy, and it's super conserved, found in flies, mammals, us. Shows how fundamental it is.
Okay. ATG4A, what does it do?
It actually has two jobs. First, it does an initial cut on the AG8 protein, getting it ready. That part doesn't seem to depend on the redox state.
Okay.
But its second job is crucial here. It performs a redox-dependent delipidation of AG8.
Delipidation, taking the lipid modification off at G8.
Exactly. AG8 needs that lipid suit to help build the autophagosome. AG4A normally takes that suit off later, recycling AG8. But this delipidation step is sensitive to the redox environment.
Okay. So how does the oxidizing shift affect this?
The key insight is that under these oxidizing conditions, a specific part of AG4A, a cysteine residue at position 102, CS102 gets oxidized. And when CYS102 is oxidized, it selectively shuts down AG4A's ability to remove the lipid suit from AG8.
So AG8 stays dressed in its lipid suit.
Yep. The lipidated form accumulates because it's not getting delipidated as efficiently, and more lipidated at G8 means more autophagosome building, which means enhanced autophagy.
More recycling, healthier cells, longer life. It connects.
It connects beautifully, and they tested this directly.
Ow.
They created flies with a mutated at G4A, where that critical cis102 was replaced with a serine, which can't be oxidized, a redox inactive mutant.
Okay, what happened?
The results were stark. This single change completely abolished the increase in autophagy they saw with the catalase overexpression. And critically, it also completely abolished the lifespan extension and the improved climbing ability that health span measure.
Wow. So that single cysteine on AdG4A is the linchpin. That specific redox regulation is the mechanism for the longevity.
It really seems to be. It directly demonstrates that this redox control of autophagy via AtG4A cis102 is essential for the benefits they observed. It's not just correlated, it's causative.
Okay, so let's try and summarize this whole incredible story. You boost catalase, it quenches H2O2 really well.
Well, it disrupts H2O2's normal signaling job.
Right, which leads to this unexpected oxidizing shift in the cell's cysteine redox state,
Which the cell interprets as a kind of internal starvation signal.
triggering more autophagy, but specifically because that oxidizing signal inhibits the delipidation step controlled by AdG4A cysteine 102.
Leading to more active autophagosomes.
And ultimately, longer health span and lifespan, at least in female flies.
Oh.
It completely flips the script on antioxidants and aging, doesn't it?
It profoundly challenges that simple view of ROS as just damage. It highlights this incredibly sophisticated role of redox signaling as a modulator of longevity. It's about balance and communication, not just destruction.
And it elevates autophagy again as this central process for health.
Absolutely. Enhancing autophagy seems to be this deeply conserved strategy for survival and health. The aha moment here is realizing it's not just about eliminating bad molecules, it's about understanding the intricate signals. This opened up totally new ways to think about pro-longevity strategies, way beyond just taking standard antioxidants.
Yeah, it's a much more nuanced picture.
Definitely. And maybe here's a thought for you to chew on. If this perceived starvation, this internal redox signal, can be pro-longevity by activating autophagy, what other subtle stress signals might our cells be sending? Signals that, if we could understand them, maybe gently nudge them, could be harnessed for better health and a longer life. Makes you wonder about all the internal conversations happening right now.
It really does. What an absolutely fascinating deep dive today. We hope you enjoyed unpacking these really surprising findings with us. If you found this deep dive as compelling as we did, please do like this video and subscribe to stay informed on the most current and impactful research. This has been The Deep Dive, part of the SANS Science Experience.